be on the paracetamol half-life, which is basically an extension of last week's practical. Before I go through the protocol for the last practical, I just wanted to give some feedback and some advice on the practicals so far. So most of our practicals to date have involved standard curves, have involved making stock solutions, making standards, making samples. So what's most important to get right here is pipetting and mixing. So check that your pipetting is okay. So how do you know this? Well, you can have a look at things like the optical density of values of your standards and samples. Make sure that they're precise, that they're all very, very similar, and that they're close to less than 1% of the particular value. If they're higher than others, it is likely that you're not pipetting correctly. In terms of other explanations, there's a possibility that you've made the stock solution up incorrectly. There's a possibility that you haven't been doing adequate mixing. So that means every time you add like a chemical to your standard, or that you've mixed or diluted your standard or sample, that you haven't given the requisite time to mix it properly, either by vortexing or by pipetting up and down. You need to make sure as well that your calculations are correct as well in terms of calculating standard deviations. And I suppose mathematically you can actually check to see that your pipetting is precise. You can check your coefficient of variance, your CVs, and make sure that they're less than 1%. So this, I suppose, is ultimately what we'll be checking during the exam, and when you submit your final version of your exam paper, your practical exam, we'll be looking at the percentage CVs for both your standards and your samples. So if you do have any questions, or you're unsure still about whether or not your pipetting is accurate or precise, please do ask at the next practical. So for the last practical then, we'll be doing the paracetamol assay again, same as last week, but this time instead of having one unknown, you'll have 9 unknown samples. Okay, and the idea for this is to be able to generate a plasma concentration curve so that we can estimate the half-life of paracetamol. So there's no need to dilute them. You're going to read the standards and all the samples together on the same plate. You're going to plot the standard curve as before with your standards. Make sure to subtract the zero, the blank. Get a straight line through the origin. Estimate then the sample values from that particular standard curve. You're then going to divide them by 200 to get the actual true value, because they've been concentrated by that much. And then on semi-log paper, you're going to plot the values versus time. And finally then, using the linear part of the graph, so the last part, 4 values usually, you're going to find the t1/2. So you pick a concentration, find the time of that time at that particular concentration by just doing a extrapolation, and then pick half that concentration and find the time at that concentration as well. The difference between the two values is the t1/2. So you'll be used to seeing graphs like this, a plasma concentration curve, where you have the concentration on the x-axis and you have the time on the y-axis. And normally with an oral drug, at least, you see an absorption, you see an increase in concentration until it reaches its Cmax, its maximum concentration in the plasma, at Tmax, the time it takes to reach the Cmax. And then through a number of different processes, usually by metabolism and by elimination, the concentration decreases and eventually goes down to 0. We want to find out, obviously, these types of parameters are very, very useful pharmacokinetically for determining dosing regimens, from ensuring as well that the drug maintains itself between the therapeutic range, between the minimum effective concentration and the maximum toxic concentration. So for this, we need to find out things like the t1/2, and as I said to you, you're going to be plotting your concentration. You get the log concentration on semi-log paper, and you're plotting it against time. So you pick two concentrations, and you get half of the initial concentration, and you find out what time each of these are corresponding to. And the difference between them is the t1/2. Okay. For the next part of the practical, so that is practical 6 basically finished, but then there's also an extra exercise as well that's linked to it. So you'll submit practical 6 as is in your lab manual book, but then there's an extra part to it that has to be submitted before the exam the following week, and you're going to use your own results then to do that particular exercise. So you'll be calculating things like the elimination constant, shown here on the graph. You'll be looking at the area under the curve, and you'll be also getting some other constants as well. Okay. Before that then, as I said, you have to submit that before the exam, before the actual lab exam. There'll be a separate submission link for that. Using your own results, you'll calculate the elimination constant, you'll calculate the half-life of absorption, you'll calculate the absorption rate constant by the method of residuals, and you'll calculate the area under the curve as well. Okay, and you'll use that then as you'll use your results from the half-life of paracetamol assay to do this particular exercise.